So now I want to discuss about the essential functional unit of living organism, which is called protein. Then how the protein serve their function. And that secret lies in the structure of the protein. So we have studied protein structure for many decades, and we figured out how they actually is composed of, how they look like, and how they serve. So for that, here comes a simplified version of, you see, we studied amino acid can be as a 3-letter. So this ASN, glycine, phenylalanine, glutamine, like this. So this polypeptide sequence of this amino acid, we call this as a primary structure. So the primary structure is the sequence of a chain of amino acid. So that's a basic building block. And however, if you look at it more closely, they actually form not just it's a linear, but it's not like a line. In real 3D space, they actually form some structure. You see something like this, very interesting structure, and this and this. Of course, this is cartoon, but this is an insight to we understand how they actually look like. It's a regular substructure. So this we call as a secondary structure. That is, sequence of amino acids are linked by hydrogen bond. You remember amino acid, the one side of the carbon atom is always hydrogen. So there's hydrogen, and this will form hydrogen bonding to give us this secondary structure. So people realize there's this helical structure named as alpha helix. And the other is this part, this is called pleat, which is like in your some clothes or skirts, you see this pleats, and that we call beta pleated sheets because it looks like sheets. In fact, this clarification of the structure was discovered and clarified by Linus Pauling at Caltech, and he got Nobel Prize in 1951 and he named for this. And this secondary structure is a substructure, and the whole full 3-dimensional structure, it looks like this as an example, as a 3D structure, and we call this as a tertiary structure of protein. So that should be a 3-dimensional arrangement of these two secondary structures with one single polypeptide chain. So this single polypeptides will have an N-terminus and C-terminus, and from start to the end, this is a single polypeptide chain. So we call it tertiary. And there could be even more complicated protein which will have these tertiary as a subunit and multiple subunits put together to make one fully functional protein. So that we call as a quaternary structure, because there are a very complex protein molecules which will have multiple of these tertiary structures. All polypeptide within a complete protein with multiple subunits. A good example is a hemoglobin protein molecule, which has like 4 subunits, and this will carry hemoglobin, oxygen in our body. So it's a red molecule, one of the most important functions in our body, especially blood. So that protein structure and function, let's get into a little bit deeper. How the primary structure, as I alluded to show you, like how the peptide bond each other, and then bonding makes a polypeptide chain. So each amino acid will be represented as one residue R1, and you can see R1, 2, 3, 4, 5, and that will have one side eventually is an amine, and the other side is carboxyl COO minus, you can see. And this carboxylic acid, because it's C, we call it as a C-terminus. And the other amine side, you see this N, so we call it N-terminus. So this polypeptide chain always has one side N-terminus, the other side is C-terminus. Interestingly, this sequence, when it's inside a solution or water, and it actually forms a three-dimensional structure that we call as a secondary structure. You can see this helical shape. You realize there's oxygen here. The other side, there's the hydrogen. So you see this hydrogen, hydrogen, hydrogen is partially positive and oxygen partially negative. So there will be electrostatic interaction, attraction. So they will form this weak, but many this bonding will form, will become very important to make this form alpha helix structure. So this we call as a hydrogen bonding forming. And in other cases, you see this N to C, this C to N terminus direction, and this C to N terminus direction. You can see one side change of hydrogens can interact with the other side of the oxygen, and this forms an hydrogen bonding and three-dimensional structure looks like pleated sheets. So that we call as a pleated sheet. And this will form a substructure of protein, and that one single polypeptide chain, in fact, can create this three-dimensional structure as a one single amino acid chain. So that's called tertiary structure, and multiple tertiary structures can form a full protein, which is fully functional one. And in that case, we call it quaternary structure. So it's a home exercise. Why don't you take a look at this hemoglobin molecule and delve it into its full functions. So we discuss about the basic principles of proteins, and which is polymers of amino acids, and how this diversity of about 20 natural amino acids influences its three-dimensional structure and its function. Next time, we'll discuss about nucleic acid. Thank you.